Okay, hello everyone. Today we're going to look at how to use a microscope, and I'm going to show you parts of the microscope. I'm going to show you how to carry it safely, and I'm also going to show you how to look at something, look at a specimen on the stage. Okay, so to start with, let's talk about how to move with it. You're going to have to collect it from a trolley normally, so we're going to have to hold it at 2 points on the microscope. So it's always a two-handed thing that we're going to do to walk with it. The first one is to have your hand underneath the base. Okay, so this part of the microscope here is called the base. And the second hand is always going to be wrapped around the arm. So this part of the microscope here is called the arm. Okay, so whenever anybody's walking with a microscope, any teacher will expect you to walk just like this. That's the safest way to walk. Obviously, mine's already plugged in. It won't be plugged in when you're walking with it. Okay. Also on the trolley, you will have a cable that plugs in to one of the electric points around the classroom, and it just plugs in, well, this one here just plugs into the back there. Sometimes you might have a little switch on the back. My switch is on the front, and we can see that the light turns on and off like that. Okay, now parts of the microscope. A lot of the microscope here is very expensive, so we have to be very careful with a lot of it. To start with, at the top, we've got something called the eyepiece lens. Okay, and it's called the eyepiece lens because that's the bit that we look through. Okay, that's the bit that the eye goes to. Further down from the eyepiece lens, and the eyepiece lens swivels round. So at the minute, we can't share microscopes, but if somebody else was to look at your microscope, without moving it, you could just swivel it round for somebody else to look at. Further down is our second set of lenses. Okay, these are called objective lenses. And these ones are nice and easy because the bigger the lens, the bigger the magnification. So the bigger the picture you'll see through the eyepiece lens. So to start with, I'm going to select the smallest eyepiece lens. And it clicks into position. But we'll talk about that in a second. When we look through then, we've already found the light that's down the bottom here. So that's the light. You can either have an electric microscope where you get the light as a torch, or you can get a mirror and you might have to shine a lamp on the side and then direct the light up using the reflection from the mirror. But both work really well. And that shines a light up through this big thing here that's called the stage. Okay, this stage is where we're going to put our specimen so we can see it. So my specimen is here. They're very, very brittle, very thin glass slides, and you probably won't be able to see it on the video, or you might be able to, but you can see just a little bit of, this is a plant specimen on that slide. Okay, so that's what we're looking at. It's a very thin bit, and that goes underneath things called stage clips. Okay, so I'm going to put my stage clips on the top, and I'm going to arrange for that specimen to be right above this glass hole so I can see that the light's coming up through it. Now the last two parts of this microscope that we should have left to label are some focusing wheels. Okay, so we have firstly a coarse focusing wheel, and you'll see when I move it, it moves really quickly. Okay, so this is like a rough focus that we can spin, and then underneath it is called a fine focusing wheel. So if I spin this, probably on the video, you can't see it move, well, hardly at all, even though I'm turning it and turning it and turning it. If I carried on turning it, it would go all the way down to the bottom compared to the coarse wheel. So a quick recap, our eyepiece lens that we look through goes down to our objective lenses that spin and get higher magnification the bigger the lens is. It's written on the side as well, so you can see the magnification. Down to the stage here, where we place our specimen, our glass slide, underneath the stage clips that we can see that hold it in place. Underneath we have our light source that can get very, very hot, so we've got to be careful with that. And when I look through, I'm going to use my coarse focus wheel and my fine focus wheel. Okay, so how do we use a microscope then? Well, it's very simple. We're going to use a few steps, and it's the same every single time. Doesn't matter what you're looking at, it's going to be the same. So I've selected the smallest objective lens. Okay, so here, this one is 4 times magnification because I don't know if we can see on the camera here. Can you see it says 4? All the students in my class are going, yes, but it's backwards. But on the video, it shouldn't be. So that's 4 magnification. Then we've got this one that's 40. And then if we can see the yellow one, oh, we can't because it's backwards. It says 10. The yellow one says 10. So I'm going to select my 4 magnification. That's clicked into place. I'm going to move my coarse focus wheel all the way to the top. So that's all the way it will go. I'm going to look down the eyepiece lens and I'm going to adjust with the coarse wheel until I can see an image. So I'm going to keep going until I can see something. Perfect, there we are. Now, you'll find it's very, very light. It's very bright, it's very faint. So you're really going to have to go slowly with the coarse wheel. But it's important that I put the stage at the top and then I move the stage down every time. Okay, once I've got that, I'm going to not touch anything apart from my objective lenses. I'm going to increase the power of my lenses. So now I'm seeing in a much higher magnification, but the magnification is just a little bit off. So I'm now going to use my my fine wheel just to fine tune and get my picture back. Let's find it. There it is. Now, at this, I can see individual cells. I can see individual nuclei that are in the cells. And I could go one step further and go to my highest magnification and use my fine focus wheel one more time to really focus in and really magnify my image so I can see bigger images of cells. This magnification lens is the most tricky to work. So you might find that you are perfectly happy with the middle magnification objective lens, which is 10. So I'm just going to get my picture back. The last thing is to work out our total magnification. And all we do is we times the numbers on both of our lenses together. So here it says 10 times, we can see on there, it says times 10, that little bit there that I've left uncovered. So this eyepiece lens magnifies everything 10 times, and the yellow one is also 10 times. So my total magnification is 10 times 10, which is 100. So what I'm seeing here is 100 times bigger than is actually on the stage. And that is how we use a microscope. Hopefully, we know how to walk with it safely. We know what all of the different parts of the microscope are and what they're used for. And we know the step-by-step method on how to see a specimen under the microscope.